Assalamu alaikum and hello everyone. I am Madam Michelle Mariam from KMM. Today we are going to learn how RT-PCR is used to amplify genes from RNA. Let's begin. Before we look at the steps involved, let's first understand what RT-PCR is. RT-PCR stands for reverse transcriptase polymerase chain reaction. It is a technique used to amplify genes from RNA molecules. In this process, messenger RNA, or mRNA, is first converted into complementary DNA, also known as cDNA, through a process called reverse transcription. Once the cDNA is produced, it can be amplified using the polymerase chain reaction, or PCR, to generate many copies of the target gene. In simple terms, RT-PCR involves two main stages. First, converting mRNA into cDNA, and second, amplifying the cDNA using PCR. The first step in RT-PCR is reverse transcription, where mRNA is converted into complementary DNA, or cDNA. To carry out this process, several materials are required. First, we need the mRNA strand, which contains the genetic information to be copied. Next, DNA primers are used to provide a starting point for DNA synthesis. The enzymes required are reverse transcriptase, RNase and DNA polymerase. Lastly, we need DNA nucleotides consist of adenine, thymine, guanine, and cytosine, serve as the building blocks for the new cDNA strand. All these components are mixed together in a reaction tube before the reverse transcription process begins. Let's look at the reverse transcription process in RT-PCR. First, an mRNA molecule is isolated and serves as the template. Next, a DNA primer, usually a short poly-DT primer, binds to the poly A tail of the mRNA. Once the primer is attached, the enzyme reverse transcriptase synthesizes a complementary DNA strand, or cDNA, using the mRNA as a template. This results in the formation of an mRNA-cDNA hybrid molecule. The original mRNA strand is then removed by the enzyme RNase, leaving behind a single-stranded cDNA molecule. Another primer binds to the cDNA strand and provides a starting point for DNA synthesis. DNA polymerase then adds DNA nucleotides to synthesize the second DNA strand in the 5' to 3' direction. As a result, a double-stranded cDNA molecule is produced. This double-stranded cDNA can now be used as the template for amplification during the PCR stage. After the reverse transcription step, double-stranded cDNA is produced and serves as the template for PCR amplification. Before PCR can begin, several materials are required, that is double-stranded cDNA, DNA primers, Taq polymerase, and DNA nucleotides. All of these materials are added into an Eppendorf tube and mixed together. The tube is then placed into a PCR machine, also known as a thermal cycler. The PCR machine automatically changes the temperature according to the program's cycles, allowing DNA amplification to occur. Let's see how PCR generates millions of copies of this cDNA. PCR consists of three main steps that are repeated for multiple cycles. The first step is denaturation. The reaction mixture is heated at high temperature, about 93 to 98 degrees Celsius, causing the double-stranded cDNA to separate into two single strands of cDNA. The second step is annealing. The mixture is lowered to 48 to 68 degrees Celsius, allowing primers to bind to their complementary sequences on the cDNA strands. The third step is extension. The temperature is raised to about 72 degrees Celsius. At this temperature, Taq polymerase adds free DNA nucleotides to the 3' end of DNA primers and synthesizes new cDNA strands. One complete cycle produced two new cDNA molecules. These three steps, denaturation, annealing, and extension, are repeated many times. As the cycles continue, the amount of cDNA increases exponentially, producing millions of copies of the target gene. Let's recap what we have learned today. RT-PCR is a technique used to amplify genes from RNA. The process begins with reverse transcription, where mRNA is converted into cDNA. The cDNA is then amplified by PCR to produce many copies of the target gene. Understanding RT-PCR is important because it is widely used in molecular biology, genetic research, and disease detection. Thank you for watching, and I hope this video has helped you understand the RT-PCR process more clearly.